Hey everyone, welcome. If you're tackling section 2 of D82, you are in exactly the right place. We're going to cut through all the noise and focus on the core concepts you absolutely need to know. We'll build everything up from the ground up so you can walk away from this feeling ready and confident. All right, let's get started. So here's our game plan for today. We're going to build our understanding piece by piece. First up, the classic rules of inheritance. Then we're going to zoom way in to see the molecular machinery that makes it all happen. After that, we'll look at what happens when errors, typos, creep into the code. And finally, we'll see how those tiny errors can drive the massive process of evolution. All right, section 1, the blueprint of inheritance. Everything starts here, right? With these simple but honestly super powerful rules that Gregor Mendel figured out just by looking at pea plants. Getting this down is the key to understanding everything else that follows. So what exactly is Mendelian inheritance? Well, you can think of it as the original rule book for genetics. It's the set of basic principles that tell us how traits get passed down from parents to their kids. It's really the why behind why you might have your dad's eyes or your mom's smile. Okay, this slide shows a really key power dynamic. A dominant allele, it's like the loudest voice in a conversation. If it's there, it gets heard, it gets expressed. The recessive allele is way quieter. You only get to see its trait when there are two copies of it, with no dominant one around to overshadow it. It is all about that masking effect. Here's some vocabulary you just gotta know. If an organism has two identical alleles for a gene, we call that homozygous. Just think homo for same. But if the alleles are different, it's heterozygous, hetero for different. Getting these two terms straight is absolutely crucial. And this right here, this is the huge difference between what's written in the code and what you actually see. The genotype is the genetic makeup itself, the specific instructions, like PP. The phenotype is the physical result of those instructions, the thing you can actually observe, like having purple flowers. Okay, let's move on to section 2. So we've talked about Mendel's rules, but how do our cells actually follow them? Now we're going to zoom way into the molecular level and look at the actual machinery that reads that genetic blueprint and brings it to life. This whole operation runs on a 3-step process, and it's so fundamental it's called the Central Dogma. First, the cell needs to copy its DNA perfectly. That's replication. Then, it makes a temporary, portable message from that DNA. That's transcription. And finally, it uses that message to build a protein, and that, my friends, is translation. So just remember, copy, transcribe, build. So who's the hero of replication? It's this enzyme called DNA polymerase. Think of it like a master builder. It just zips along the DNA strand, grabbing the right building blocks and locking them into place to create a brand new, perfect copy. Now, for transcription, we have a different worker on the job, RNA polymerase. This one is more like a scribe. It doesn't copy the whole library of DNA. It just goes to a specific gene and writes down a portable message from it. And that message is what we call RNA. Last but not least, in translation, we meet tRNA, which stands for transfer RNA. This little molecule is basically the delivery truck. It reads the RNA message 3 letters at a time, grabs the exact amino acid that matches that code, and hauls it over to the ribosome to be added to the protein chain. Pretty cool, huh? So we've got this beautiful, elegant system for copying and reading our genes. But it's not always perfect. Now we get to the part of the story with a little conflict. What happens when there are errors in the code? And this really is the critical question. What happens when a mistake is made? Even a single typo in a genetic sequence that's billions of letters long can have enormous consequences. We call these mistakes mutations. So most of these small-scale mutations fall into one of 3 buckets. You have a point mutation, which is just a simple 1-letter swap. Then you have insertions and deletions, which are a little more dramatic. That's when letters are actually added or taken away entirely. Let's just take an insertion for a second. This is where extra DNA bases get shoved into the sequence. Now, it might not sound like a big deal, but it can create this massive domino effect, which brings us to our very next point. And this is the huge consequence. Because the cell reads the DNA code in groups of 3, like 3-letter words, an insertion or a deletion causes something called a frame shift. It's like adding a random letter into the middle of a sentence. It just shifts every single word after it, turning the rest of the message into complete nonsense.
and that is often catastrophic for the protein.
So now, this is where we zoom all the way out to the big picture. These tiny errors, these mutations we just talked about, they're not always just mistakes. They are the raw material, the actual fuel for the engine of evolution, driving change on a massive scale over millions of years.
Of course, the most famous mechanism is natural selection. This is the process where the environment basically selects which traits work the best. So if a mutation gives an organism an advantage, maybe better camouflage, that individual is more likely to survive, reproduce, and pass that awesome new trait on.
But hey, it's not all about survival of the fittest. You've also got genetic drift, which is basically evolution by pure luck. It's just random events causing certain genes to become more or less common. And it's a huge deal in small populations where chance plays a much bigger role.
Now, genetic drift usually pops up in two classic scenarios. The founder effect is when a small group breaks off and starts a new population. Just by chance, their gene pool might look really different from the original group. Then there's the bottleneck effect. That's when a population gets wiped out by something like a natural disaster. The survivor's gene pool, again, just by pure luck, is what shapes the future of that species.
So how do we know all this is true? Well, the evidence is just so powerful because it comes at us from all these different angles. You've got the fossil record, which is like a history book written in stone. You have comparative anatomy, which shows us the shared blueprints between different species. And now molecular biology lets us read the DNA code itself, showing us the family tree of life at the most fundamental level.
And that really brings us full circle. We started with Mendel's simple pea plants and ended up at the engine of evolution itself. The main thing to take away is that it is all connected. Simple inheritance rules are carried out by complex molecular machines. Typos in those machines create the variety that natural selection acts on. It's a process that is happening right now in every living thing, constantly writing the next chapter of life on Earth.
I really hope this breakdown of Section 2 helps you feel more prepared and a lot more confident. If you found it useful, do me a favor and hit that like button. And be sure to subscribe for more WGU study guides just like this one. Thanks so much for watching, and good luck on your exam.